All right, it is 11 o'clock. So welcome everyone to our sixth diverse webinar. This is the final of our 6 webinars we've done where we've had our theme leads from Diverse give overviews and updates. So before we begin, I'm just going to start with a land acknowledgement. The University of Alberta respectfully acknowledges that we are located on Treaty 6 territory, a traditional gathering place for diverse Indigenous communities and peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to influence our vibrant community. So I'm going to let our speakers take it from here, but I'm just going to introduce them first. So our first speaker is Dr. Isabelle Aubin. She's a research scientist at the Great Lakes Forestry Centre, which is part of Natural Resource Canada's Canadian Forest Service. And Isabelle is the Theme 1 lead of Diverse, and she's the last theme lead presenting, but definitely, I'm forgetting a term, whatever. She's the last, but definitely not the least. That's the term. And our second speaker is a postdoc with the Diverse Project who works with Isabelle. She's a forest ecologist, Morgane Den Donker. So I'm going to let you two take it away. If anyone has questions, just put them in the chat. And at the end, we'll get to a Q&A. You can put your hands up, but just put things in the chat for now. And I will disappear into the background. And Isabelle, you can take it away.
Thanks, Catherine. Good. So bonjour tout le monde. Hi. So you see my screen right now. I think it's good. Okay, good. So yeah, happy to be there. Yeah, we're a bit the last but not the least, as as you were saying, Catherine. So I will present you today an overview of the Diverse Theme 1 on forest vulnerability to global change. We'll give you an overview of how we plan to work to refine prediction of forest vulnerability, make ecological data available for modeling, trait data mostly, and to develop also and how we want to work to develop tools to support forest management. So we know that with climate change, models predicts an increase in the frequency, intensity, and duration of stressors like drought, forest fires, pest outbreaks. We know that this will lead to an increase in forest disturbances. And we know that at the same time, our forests will experience altered growing condition, mainly increased temperature in the winter and changes in the precipitation patterns. The last few years, even our most pessimistic models have been surpassed by some of the reality that we lived in some area of Canada. And Canadian and also the scientific community is very concerned. Trees are dying in large numbers. There is mortality that is observed here in Canada, but also throughout the world. There are some regions like California, in Germany, in Australia. There is some region that are transitioning from forest cover to a more open savanna type forest, and this is worrying. It is less spectacular in temperate region like in the eastern Canada, but we are seeing and we are predicting reduction in soil moisture. Some predictions say around 20 to 40% reduction in soil moisture that are predicted by 2080. So we know that these phenomena will occur. We know they are occurring, but we still know very little about the consequences on our forest. The big question remains is how vulnerable are our forests to these stressors? So to answer this question, we need a complete assessment of forest vulnerability that include all of the 3 main components of vulnerability. We can describe vulnerability with these 3 components: the sensitivity of a species, which is the degree to which a species might be affected by an environmental change, the exposure, how the amount of change that will be experienced by a forest or an individual, and its capacity to adapt in response to this change. And to get a comprehensive assessment of vulnerability, we should quantify these 3 components. But until recently, vulnerability assessments have been predominantly based on exposure to climate. We use biophysical data such as temperature, precipitation, which are usually more available at broad scales, the scales that is needed to conduct such assessments. But the assessment of the other components of vulnerability, the sensitivity and the adaptive capacity, requires the integration of ecological data and genetic data, and this is harder to do. because ecological data are typically measured at local scales using a wide variety of methods, which the data are good to integrate and the same thing for genetic data as well, which is also very challenging, notably because of the size of the data sets, which are enormous, and also the scale that the geneticist works that is so different than forest management. So here, I just thought I will go with a concrete example of what I'm seeing here. I'm saying here on how the inclusion of ecological data affect our capacity to assess forest vulnerability. Here, the example is with sugar maple in the eastern Canada. So using climate data and based on where the species are currently located, we are able to project where the suitable climate for a given species will be in the future. This is what we call their climate envelope. And so this is it gave us information about the exposure of to climate change. So this map shows on the left in green the current distribution of sugar maple in Canada, and in blue the projection of its climate envelope by the end of the century. That's what I'm speaking about, the exposure. And what I'm presenting here, just to say, it's under a high carbon emission scenario, just to let you know. So climate envelope tell us where the climate will be favorable to the species in the future, but it does not tell us about the species response to this change in climate. Will the sugar maple be able to adapt to the new growing condition where it grows, or will it be able to migrate and track its suitable climate? Climate envelope does not tell us this information. To get a sense of the tree ability to migrate, we have to integrate ecological data on sugar maple migration capacity. We can think here of seed production, seed dispersal rate, the age at which a species get maturity, sexual maturity. And so you need to integrate this information into the model. So we have done this exercise here. When we integrated these data, we were able to get a better sense of the capacity of sugar maple to track its climate. So in the map on the right, that's where we have integrated this information. And you can see in yellow the area that will potentially be colonized by sugar maple by the end of the century. So it clearly shows that sugar maple does not have the migration rate fast enough to follow its climate envelope. So we also see in this map, oops, sorry, I touched something I should not. Okay. We also see in this map that the proportion of the current distribution of sugar maple is projected to end up outside its climate envelope by the end of the century. So the estimation we have here, again, with the high emission scenario, is about 15% of the current sugar maple Canadian distribution. And in the right, the map on the right, this is the area in pale blue. So the maple trees that are growing in these regions will find themselves with novel climatic conditions for the species. So the potential response for maple populations in this area is either they will tolerate or adapt to the new growing condition, or they will become maladapted, more susceptible to pest and disease, and maybe will have some regeneration failure, and in the long term get extirpated. But to evaluate this, we need to integrate ecological data and genetic information about the adaptive capacity of the tree species into our model. Just using climate data does not give us this information. So assessing forest vulnerability is really a transdisciplinary exercise that involves integrating expertise from several research fields, ranging from climate modeling to ecophysiology, population genetics, and so on. So with the help of a large team of collaborators, we have been working on the challenge of informing these least known components of vulnerability. That's really the aim of the work that we are doing. So in this talk today, I will present you our approach that we are using to get an integrated, comprehensive assessment of forest vulnerability. Then we'll continue with an overview of some of the work that has been done in the past and on which we are building on in the DIVERS project. And this will lead us to showing you our plan for the next 5 years within the DIVERS project. And we'll give you a presentation of the structure and content of the theme 1, and with a presentation of some early results en primeur. So let's start. So our approach. how we work. It's really to look at climate change on the lens of the ecological processes by which tree responds. As well presented in this Sally Atkins paper, three potential scenarios under climate change. They can persist through migration to track their suitable climate. They can persist also in situ through adaptation to new growing conditions, or they can get extirpated. And we need to understand the ecological processes that drives this response so we can better anticipate and mitigate the impact of climate change on our forest, and in this way inform conservation strategies and management practices. So one thing we know about species response is that some species will be more affected than others. Faced with the same stressor, some species will be favored, other more disadvantageous. And the magnitude and nature of species response will depend on the characteristics of the species. That's what we call their traits. I'm referring here to life history traits or ecophysiological traits such as seed size, seed production, which will inform about species migration capacity, like I was showing in the map earlier, or traits like rooting depth, xylem resistance to cavitation, which tell us about species sensitivity to drought. So to capture these species response, we use what we call a trait-based approach. So the strength of the trait-based approach is that it allows us to characterize species response across all forest species with minimal information. This approach is a form of comparative ecology. We are comparing a few key traits, a few key information about the characteristic of the plants across all species, rather than studying a single species, it's the line, the column, and we have the line. So the line, the comparative ecology, the trace ecology, and the line is the haute ecology, which would be to study one species and get all the information about this species. So this way, we are able to compare species with one another and to understand the underlying mechanisms of their tolerance or sensitivity. However, the indices we develop, of course, remain coarse. In a perfect world, we would have detailed ecological studies on every species to the various stressors that are linked to climate change, and then we can do amazing work. But unfortunately, we are still far from that goal. Currently, we only have a few commercial species that have been sufficiently studied. It will take likely several decades, if we get there, to have sufficient information on all three species. In the meantime, we are working with what we have, and a trait-based approach is very powerful to be able to include all the species and not just the one we have enough information. So our journey in this work started 12 years ago with a multidisciplinary working group that was tasked to develop a framework to facilitate the collation of trait data to be used in models of forest vulnerability. With the help of this working group, we identified traits and mechanisms behind tree sensitivity to the main climate change stressors. So to do so, we used a mechanistic workflow. For each stressors, we defined the main strategy and mechanisms that underlie tree responses. And then for each stress strategy, we then defined which traits mediate these responses and then document the relationship between the trait and the mechanism. Then from this list of traits and this information about the relationship between the trait and the function, we selected a short list of traits that capture the species response while being realistic to document, that we are able to find enough information on those species. And then based on this framework, and after a few years of data mining, because it's a long work to get all this information collated, we developed a first series of indices that was characterizing the sensitivity of major tree species in Canada for few stressors. So these indices were built around the main mechanisms that was explaining the species response, and they were very tailored for each stressors. So here in this slide, you see an example of the indices, the series of indices that we developed for drought sensitivity. So it's for the three main mechanisms: avoid drought, resist to drought, and recover after drought. And each of those mechanisms was quantified This is the red boxes using a specific traits for the tree species. So we documented the trait value of all the species, and then we were able to develop the indices with this information. So in this slide, you can see the dark red indicate a higher sensitivity. And then you can see that we have a range of sensitivity across the species we were documenting here. And also a range of also a difference between species between the different mechanisms. So a species can be good at avoiding drought, but less good at resist to drought, for instance. So these indices were designed to be easily updated as new information become available. And to do so, we were also very important for us was to present the quality of the available information in the table by adding a confidence level. So that's the column in blue you have here. So this column is very important to highlight the knowledge gaps and help to guide future research. And the priorities of the diverse theme that we'll present you later, we really we develop them based on this knowledge gap scan. And then based on these indices and in collaboration with the Forest Gene Conservation Association, we developed a series of online tools where you can explore the different indices and the processes underlying tree species response to climate change. So we have indices for stressors like fire, drought, and climate change velocity. Then we integrated these indices on tree species sensitivity with some information about the exposure of the species with the climate scenarios. Here, the example we're giving here is for drought. And also with we combine this information with stand composition. And then we were able with that to provide a special assessment of the potential impact of these stressors on forest. So we used here a color code defined by the intersection of two axes. So you can check the legend color. So in the x-axis, we have the amount of tree biomass that will be exposed, that are projected to be exposed to the stressor. And then so this is the green to blue. And then you have the y-axis that is giving the average sensitivity of the species making up this biomass. So with that, we're able to identify some area where we have hotspot of vulnerability, which will be in pink. So high biomass exposed of tree sensitive to drought. And also these special assessments were also made available online. So now the third component that I told you before, the adaptive capacity, which is the least known component of forest vulnerability. Very important to note that I'm referring here to intrinsic adaptive capacity, which is the ability of an organism, a species, an ecosystem to adapt to the impact of climate change. So this is very key to assess the capacity of species to persist in situ under climate change. So if we go back to the map I showed before, we can go back to this area in pale blue. I was telling you that the fate of sugar maple, that this region in the blue pale, this is where the species will be outside its climate envelope. So the tree are projected there to grow under a new unsuitable condition for the species, or at least new condition for the species. So we typically mention for such, when a species get out of its climate envelope, that they mentioned there will be a risk of maladaptation, risk of regeneration failure, increased mortality. But in fact, we don't really know what will happen. Will they adapt to new growing condition? We do not know. To have a bit more of a knowledge on that, we need to integrate information on the adaptive capacity of the species. So, but we are not good to do so because this is really on dirait en français, entre deux chaises. So it's being at the crossroad of ecology and genetics. So two disciplines that are not used to work together. Just take a bit of water. So we decided to get into this challenge and work at the crossroad of ecology and genetics. And we developed a framework describing tree adaptive capacity with the help of Nathalie Isabelle. That was a great a very interesting project of exchanging information between disciplines that are not used to work together. I really enjoy that work. So this framework articulates the wide range of processes involved in adaptation under two major axes. So there is the spatial and the temporal. So the adaptation go from individual short-term adaptation through phenotypic plasticity to genetic exchanges between population for long-term evolutionary adaptation. So from this framework, we develop a first series of quantitative indices of the adaptive capacities of Canadian tree species. So again, the same structure in dark red is where you have the more sensitive, not sensitive, sorry, the lower adaptive capacity. So these indices are based on life history traits related to fecundity, intraspecific trait variation as well, but also on genetic information such as genetic diversity and the level of genetic differentiation. The blue column again here inform us on data availability. And as you can see here, there's still a lot of uncertainty because more blue pale, it means we have low information, and gray boxes means we have no information available. Okay, so what is next? So this is really a work in progress. That's very important, and they have been really built that way to just thinking that we need to update the information and improve and refine the work with new data and knowledge that comes available. And when the Diverse project, when Christian arrived with Diverse project, it was just a timely for the development of a next generation of indices and tool for this work. So the theme one of Diverse is really designed this way to really bring all the information to be able to get a step for a big step further, building on the work we've done, but getting very stronger in the capacity to assess vulnerability. And the way we design it, it's very designed on making fundamental science to feed applied science. And so that's very key on the structure. We'll show you in the next slides. And also this is very based on collaborative science. There's so many people bringing expertise to this work. In the same way, it's also just in a structure. This work is collaborative as well as it's jointly funded through the Diverse NSERC and the Climate Change and Adaptation Program of the Canadian Forest Service. So it's very, we put all the expertise, the funds together to get a step further, a big step further, let's say. So the theme one, our first key goal is to generate new knowledge to fill critical gaps that we previously identified. So gaps of knowledge in tree response to climate change in Canada. So it's very important to make available the needed data that we'll need to refine the model that we'll use to forecast for vulnerability. But also it's really structured in a way that we will feed other themes of the Diverse project as well. So the project start, and really an important piece of the project is in-depth research that is conducted on a small number of tree species to elucidate some key aspect of relationship between the drought and hydraulic traits, and also between regeneration failure and root traits. Then also another critical knowledge gap that we need to fill is trait data that are often trait data set that are incomplete or geographically biased. Sorry. So we therefore work at filling gap of knowledge on seed and wood trait variation across species distribution. So a first step of the team one was a collaborative field data gathering across Canada, the Fund Tree campaign, to improve trait data coverage. That was very important, and Morgan will present you more details on this soon. And then from the collation of all this information that we will generate, all this new knowledge, and also also in the same time, we are doing extensive data mining of traits data. With all this information collated, we plan to update our tree sensitivity indices and also develop a new set of indices on regeneration failure. We aim also to increase the number of species documented. That was around 30 to 40, depending of the stressor that we've done in the past. And now we're aiming more around 90 species, tree and shrub species. And also, we are also adapting some indices on susceptibility to pests to the Canadian context. For this, we're not using a trait-based approach, as the relationship between the insect and its host is too specific to be accurately predicted by traits, but we developed the indices using a same structure, a mechanistic workflow, so they can be comparable. We can make some comparison between the different stressors. We'll show you later. So after updating these different layers of information, we will perform new spatial assessments that combine biophysical exposure estimates, so the climate projections, with sensitivity indices to create vulnerability maps, a new generation of vulnerability maps. We also plan to work on the synergies between the different stressors to improve the accuracy of the prediction, because it's not in silo, these stressors. And also, we want to continue the work we were doing in collaboration with geneticists to include some aspect of adaptive capacity. This is very important to refine our projections. So from there, we plan to develop. I think that's a key pieces. That's very the most important end product. We want to develop a new generation of visualization tools. We hope that could help stakeholders to visualize the risk and inform adaptive management options. And also, we're very happy to have this large, diverse network of practitioners that are involved. And so we hope to use this network to co-design and beta test the tool that will be developed. The map will be developed under a same straightforward and reproducible mechanism data flow that will allow us for an easy comparability between the impacts and the regions. So it makes possible to update results as new data become available. So that's very useful, but it's also for forest management, for the users that can tailor the content of the map to examine the global change impact to the specific case of their region. So if one mechanism is important, or one type of stressor or a mix of stressor, you can really tailor to the need. That's what we hope. So for instance, just giving you here two maps we had, you can see just the fact that they are produced in the same approach, with the same structure. You can observe the vulnerability area depending on the stressors, and it helps to do some comparison. So this map can be used also. We hope that this map will be used to support decision making, can also be useful to identify a region of special concern, so the hotspot of vulnerability, can also be used to prioritize areas where intervention is most needed. And then at a more regional scale, the map can inform adaptive management options, such as do we do stand conversion? What should be the good strategy for that? The species selection. And also I think it's interesting also for an adaptation perspective. It can be used to identify some region where the local population might be already exposed to extreme environment, and then we could be interested to study in this area the local acclimatization and the selection that was there, or select population from there. Anyway, there's a lot of things that we can discuss that can be done with this work. So okay, so for the end of the presentation, we thought we will give you a quick presentation of two projects that are enough advanced to show you some of the results. So this is very brand new results to show you. We'll leave the floor to Morgan, and then yeah, so your turn, Morgan.
Okay, thank you, Isabelle. So as Isabelle said, the first step was to assess some traits across Canada. So this is the Fun Tree project, the Fun Tree campaign. And the overall objective was to really document the interspecific and the intraspecific variation in wood density across Canada. So if we want to study intraspecific variation, that means that we needed to sample in multiple sites across the distribution range of species, because there is no unique trait value for one species. There is a range of trait values. Traits can vary across environmental gradients, and we have large environmental gradients in Canada. So that is why we build this project as a collaborative one to be able to get samples from multiple locations at the same time. And during summer 2024, more than 40 teams sample wood cores across 200 sites. And we collected like 1,000 wood cores on the 18 tree species that you can look at the screen. And collaborative science is actually harder than it sounds. So it's much easier to do your own research in your own lab, but because it takes much more time and demands a really good coordination to build a collaborative science project. But what is fun is that it really brings together multiple teams in a joint effort. And today we wanted to share some field picture that the participant took while doing their field work. And we gathered them in awards categories. So we can give a round of applause for the team that collected samples at the really northern tree line. We have some other teams that gather more than 200 wood cores. We have one team that sampled nearly all the species that were on the list. And finally, we also had some teams that collected samples in the USA, and we are really grateful for that, because so that we have like the whole distribution range of species, of the target species. And we also had like, we asked the team to take a picture while they were doing field work. And this is one of the funniest team picture. So we have the team, the adventurers. We don't see any trees on the pictures. But anyway, that's what we have when we build collaborative science projects. So we thought it would be fun to share a few pictures with you. So now that we have really the data, we want to answer the big question of what are the drivers of variability in wood density among the target species. And we have divided this objective in 5 sub-questions. And the first one is intraspecific variability consistent across the species? So is it smaller or larger for some species, like you can see on the graph, or is it consistent across species? So that was the first question. The second question is, which are the traits that influence wood density? For instance, in a community, we can have a proportion of gymnosperms and angiosperms that will influence the wood density value. The third question would be, you can skip the slides, Isabelle, please. So the third question is, how does wood density vary across environmental gradients and local factors? Because we know that environmental gradient can influence the trait value, and we want to know which really are the factors that influence wood density the most. Finally, we want to know, and this is a very important question, because we know that there are data that are available, but we don't know how good are the data. So we want to know how well do the existing databases, such as TRI database, represent the intraspecific variability of wood density for the target species. For instance, in TRI database, we can have a much narrow range values of wood density than we would have if the database would take into account the species across its distribution range. And the final question is, to what extent does the measurement technique affect wood density? Because there is several methods that exist to measure wood density, And we want to know if what is the influence of the method and if the influence of the method varies across species, which would cause some bias in the data, and that would not be a good news for us, actually. So at the end, we have like several data sets that we want to compare. And you can see on the slide that these data sets do not have the same species. They do not share the same method for measuring wood density. We have a large range of values in the data sets, and also the geographical range is very different. So let's just pay attention to two data sets, the FUNTRI1 and the FUNTRI2, that were used to really test the influence of the method on the wood density. And also we have some data sets that have a really narrow geographical range, such as the ALEMDAC dataset, and compared to our datasets, which encompass the whole Canada. And so here is some preliminary result from a student who has been working on these different datasets. So first, if we compile all the datasets together, we can already see that there are some differences in the coefficient of variation across species. So we have some species that present a larger intraspecific variation than others. For instance, we have the Tilia americana who has a coefficient of variation of 15% compared to the Tsuga canadensis who present a coefficient of variation of 5%. And interestingly, we can also see on this graph that the distribution of wood density values across species, they are not the same. Actually, the shapes of the curve is not the same across species. The second result that I wanted to show you today was the difference between the two methods that were used to measure the wood density. And I want to make that clear that we use the two methods on the same samples. So there are no other factor of variation than the methods. And what we see is that we have a good relationship between the first method and the second method. And then one method, the scan method, gives a higher density than the immersion method. And there are no species-specific effect that was detected. That means that the bias, what we call the bias of the method, is consistent across species, which is actually a really good news for us. And finally, if we compare the three datasets together, there are two datasets that have a smaller geographical coverage. So this is what we get. I've hidden some lines because we do not have all the data yet for all the species. So let's just focus on two species that have a wide geographical coverage. So the white spruce and the trembling aspen. And we can see that the intraspecific variation is higher in the FUNTRI dataset. So that is the blue line. So the FUNTRI dataset shows a higher intraspecific variation for these two species. These two species have a large geographical distribution. And so this is preliminary, but it would seem that the dataset that have a smaller geographical coverage did not encompass all the intraspecific variability. This is kind of logical because we know that environmental gradient can affect trait value, but this would be a confirmation of this hypothesis. But again, this is preliminary results, so let's wait for the next. And I think that's all for the FUNTRI project.
Yes, thanks, Morgane. Yeah, so that's really give you a taste of what we are doing at the really filling the knowledge gap and trying to make sure we are able to collate the best way the data we're coming from, what we have generated and the data we are bringing from other data set in a way that is accurate. And then I will bring you to the other spectrum of the team one, where we will see how we are integrating all this information into vulnerability assessment. And this is for vulnerability assessment on exotic pests. I told you for exotic pest, we are using we just adapt an indices that was already existing. So we were able to start doing the vulnerability assessment. We were not at the stage of collating the information to update the indices like we do for the other indices. So I thought I will show you where this new maps that we finished recently that we hope to publish this year. So because we know with all the new insect introduction, we really felt that it was really important to include this stressor into our vulnerability assessment. We were very personally, I really have a question on trying to understand how much of the Canadian forests are at risk of being affected by exotic pests. You know, we hear all the time new pests arriving. And I'm really asking what will be left after all these insects have passed, and is there some region in Canada where we are more vulnerability hotspots or region that are less affected? Is there any species less affected as well? So to get a sense of these questions, we use the same approach as the other maps I showed you before. We combine the distribution of the 14 exotic pests of concern that are currently expanding in Canada and have the potential to induce a significant tree mortality. So we have this 14 exotic pests that we selected with, so we combine their distribution with the distribution of the 37 most abundant tree species in Canada. In Canada. So, and then we use also climatic scenario to project the pest species climatic niche into these Canadian tree species distribution to have an idea of where they can, how much they can expand into the landscape. And we included to our analysis, as I told you, characterization of the severity of the insect, of the infestation. So our index goes from a low severity where only mortality of weakened tree is likely to occur to a high severity where functional extension of the tree species could occur. To give you an idea, those 14 species represent a range of severity. It includes, for instance, the brown spruce longhorn beetle, which currently found on Nova Scotia, where it's causing mortality mainly on weakened or stressed spruce trees. But with the transport of infected wood, it could potentially spread and affect other spruce species in other regions of the country. There's also the beech bark disease. It's another pest that we included, which has a higher severity than the brown spruce longhorn beetle. It is currently causing extensive mortality within the beech population in eastern Canada. So this is not a new pest. It's present in Canada for several decades, but it is currently increasing its distribution in Quebec and Ontario. We also included the mountain pine beetle, which is a very good example of synergy between the pest and the climate change, with the warm and dry summers in central British Columbia and Alberta, and with the mild winters, it having lead to an increase in its population. And the projection of its climatic niche indicate it could potentially be an important threat for the pines in eastern Canada. Just maybe to mention that on the map, we excluded the historical distribution of mountain pine beetle, and we only included where it's outside is normal distribution into this work. And then we also included emerald ash borer, which is currently causing massive mortality of ash species. In the last 20 years, this insect has a rapid spread, mainly via the transport of infected wood, firewood. And we're working on this insect for the last 15 years following the impact. And this insect has the potential really to bring the functional extension of ash species with a mortality rate over 98%. So from this integrated assessment, we obtained a map combining the current distribution of the 14 selected exotic pests with the 37 most abundant tree species in Canada. So the map shows the areas where current tree species biomass are exposed to exotic pests and the weighted average severity of the insect damage. So we use here again a color code. This is defined by the intersection of the two axes. In the x-axis, the quantity of tree biomass at risk so these trees are at risk of mortality by at least one pest. And in Y is the severity index, which is ranging from green and blue to severe massive mortality, which is orange to purple. And I'm adding here on the right a second map with the projection of the potential progression of exotic pest infestation in the short term. So this map is based on the climate niche of the 14 pests. But it is important to note that projection does not constrain by the pest dispersal rate. It's only inform us on the climatic niche, just so where the host and the climate is adequate for the pest for its development. So these maps, it's useful to identify hotspot of vulnerability, which will be in pink. And we hope it can be useful to determine region that may require close supervision to limit potential impact and safeguard forest. And again, here it can be used to support decision-making and prioritization of intervention. So that was a quick overview. So I will just leave it now with time for a question, and thanks for listening. Just need to go here too.
Perfect. Thank you so much, Isabelle and Morgane, for sharing with us and for all your work in theme 1. So yeah, we'll open up to questions now. So if anyone wants to you can put your hand up or you can write it in the chat. I think we already have one in the chat I can start off with. But if you do want to speak like verbally your question, just put your hand up and then I'll allow you to speak and then you can ask her. Oh, both of them. So first question is for Morgane. Any thoughts on why you think the distribution of WD is bimodal for Larix laricina? I find that fascinating. This is from Julie Messier.
Yes, I see that. Thanks, Julie. Actually, I'm not sure that the distribution is really bimodal because we don't have all the data yet for that species in particular. So this might be due to the small number of values. We have like 15 cores analyzed for this species at the moment. So I would say let's wait for the complete data to see if it's confirmed. But that would be interesting, yeah.
Thanks, Morgane. Anyone else have questions? Please put your hands up if you want to, or type it in the chat. Maybe you both explained everything just perfectly.
Yeah, we were perfectly clear.
Oh, we have one hand up from Julie again. Julie, I'll just allow you to talk, so you should be able to unmute now.
Hi, everyone. Well, I mean, I'm just jumping in because I don't see other people having questions, but I thought it was really interesting, the preliminary analysis by Thomas, and the fact that, as we would expect, it seems like there's more intraspecific variation when you have a data set that covers a broader geographical range. So I guess I just have a few like follow-up questions, and I know this is preliminary stuff, but has he had the time to maybe see if there's a correlation between like the environmental gradient covered by that data set and the intraspecific variability, you know, like knowing that there's only 3 data points, but we'll have to parse out the environmental variation versus the spatial gradient, because you can like stretch a very large stretch of the Arctic but then stay in the same climate, right? So I just wanted to kind of hear your thoughts about that.
He didn't have any time to do that, but I don't think he will be doing that because he has to like write his report soon. But I will do that soon, I hope, as soon as I have the whole data.
Okay, thank you. All right, and I was looking forward to continue chatting about this in our future meetings.
Yeah, sure, me too.
Okay.
Thanks, Julie.
Thanks.
Anyone else have any questions or comments for our speakers? We have a quiet group.
People are just shy today.
Yeah. Well, that's okay. Yeah, if anyone has questions for Isabelle or Morgane in the future, I'm sure that you can contact them. Their contact information is on our website too, or you can reach out to me. So if you need that. And yeah, if you want to share this. Oh, we have one question from Olivier. For the Fun Tree campaign, some species with lots of observations had very wide distributions, and some with very few observations had wide distributions. How can you deal with this?
Morgan, yeah.
Yeah, that's a really good question. Actually, we won't be able to study intraspecific variation for all the species. Obviously, if we do not have enough data points for species with a wide distribution, we will not be able to do the analysis of intraspecific variation. So that's too bad. But the result that was presented here are preliminary. So the numbers of data are actually higher than those that are presented. So hopefully we will get there. But I think for the species with the widest distribution, we have quite a few data. Yeah.
Well, thanks, Morgan. You're really in the hot seat for all the questions. I have a couple hands up. Karen Diamond, I just gave you permission, so you should be able to unmute. Yeah.
Hi there. Yeah. So I'm on theme 4, which is the Landos modeling group. And I'm curious how you see the modeling being connected to these various indices and traits. Like our models don't track variability in wood density, for example.
Yeah, we are in discussion with team 2 and 3. So so we had chat with Clément. We are kind of, I think it's really, and this is something we can discuss later as well, but we are doing discussion to the best. Okay, I can tell you from how we've done in the past and what we are starting to do right now. It's really to sit, and Morgan have done that with Clément, to sit to see what are the pieces that you have in your model, and then we can say how we can bring the ecological knowledge to that we say, okay, this exists, it's possible to include into it. So that's one thing, that's one way we can do it. It's really a question of communication and taking time because we know more of the process, the ecological processes, and how we can transform that in traits. You know more about the modeling, so we need to chat and make sure the information can pass from one discipline to the other. Then another way as well is to see, and this is Clément work as well, he had done, is to see, okay, what if we include intraspecific variation or we don't include it? So then it will see how much it will be a difference that you don't include it. So that's a way you can work as well. Another way as well that we can include it is to see, we are kind of looking, there is different, like, let's think of a species that is widely distributed. You may have different population, genetic population that may have different different trait structure. So we can try to define that, and then you can use, because species, it's a concept, it's not that it's in fact it's quite a wide variation of local adaptation, and so we are using species as a defined unit, but in fact there's a lot of variation within the large distribution of our tree species. So we can try to include that as well in the analysis, and trying to have kind of an average for different population, that we know that are more distinct. So that's all different ways that we can integrate that, but it needs time, it needs discussion, and yeah. So I don't know, Morgan, if you want to add more to this question.
Maybe just maybe a few things, because you said that team 1 is really building on previous work, and so Fun Tree is actually a really small part of team 1, even though it was huge work. But your lab, Isabelle, has already really a large amount of sensitivity indices that can be used directly into models, not only wood density. I mean, you have other sensitivity indices based on other traits that can be used in models. So that, just wanted to add that.
Yeah, there's several. So I don't know, Karen, if we answered your question, because there's so many different ways we can we can get there. But I think it's all means we need communication, I think.
Sounds about right. Okay, we have a couple other hands up, and I think that'll take us to the end of our time. So Diana, I just allowed you to unmute. Yep.
Yeah, hi, great. Yeah, thanks for letting me raise my hand instead of typing. It takes too long. Yeah, so that was really interesting to see, especially because I had contributed a couple samples from the Yukon. So for the intraspecific variation in wood density, like you showed, well, for white spruce and I think aspen, it was like a wider range, but then on the lower density end, it was basically the Fun Tree data were missing like the lower range. So I was wondering if that was just like a geographic distribution thing, or if there's other differences between the data sets, like tree age or the even like the timing of the samples or anything like that. Like, how would you explain that?
Yeah, I see what you mean. Geographically, the two other data sets, we cover the same region. Our data sets covers the whole Canada, and the data set that I showed cover like one location in Ontario, actually. So for the location, we are good. There might be some bias because of the methods that was used to measure wood density. For instance, our data set, we used the scan method, and the two other data set that I presented used the immersion method, and I showed that there was a difference between the methods. So that could explain why we don't get the lower end of wood density. So this is my first thought about it, but definitely worthwhile to dig in.
If I can add also, when you will have the information, when you will do the analysis with the climate gradient as well, we can check as well if there's not kind of a, depending of, because our sampling was going with who was available where, so we can make sure it's not related.
But we have the same location. We sample at Petawawa, and the data set comes from Petawawa as well. So I would say that.
Okay, sorry.
Yeah,
I was
okay, maybe also climate change, because the data set dates back from.
That's
40
what I was going to ask, if there's any change in time over
40 years actually. So yeah.
Yeah, so I wonder if that could be something.
Maybe it's interesting.
Yeah.
And thanks for the participation to the campaign.
Yeah, for sure. Thank you.
Okay, we have one more hand up. So Marie-Josée, you can unmute now and ask your question.
First, thank you for this very overview that was quite complete and very informative. It's in the same spirit of the previous person, that the time difference that may have been confounded with the climate. You know, it's not just the same location, but the new climate may affect the condition. But to add to Karen Diamond comment, we spoke yesterday about this, is some trait or values that can be measured about the trees sometimes cannot be are not sensitive enough in Landis. So I know Morgan and Clément, you did a little experiment of which trait to behave and responds to a change of magnitude. So this is something that we need to add more sensitivity to make sure that it's not useful to add an information if the model doesn't respond to it. But on the other hand, maybe the genetic part that you mentioned, Isabelle, that with your other research group, I think that's very relevant. And one way we can address that with models is to create pseudo species. So we have a current species and a future species, and then we can replace them as the climate, the temporal dimension of simulation, as you have shown in your previous research. So I think we are all aware of this. And now soon in Team 4, we will have the student to help us to address not all of those questions, but some of them. So thank you. And hopefully one would be interested in the NSEC. So I don't know. That's important. Thank you.
I think we should really see that as a work in progress and always thinking of we refine a little piece here by breaking the silo of the different work and bringing new information where it's relevant, but it's not relevant everywhere. I fully agree with you, Marie-Josée, but it takes a lot of time and, discussion and try to see where it's the most useful, but then we can I think it's important so we can improve and refine the work. So, but I agree with you, not everywhere. That's very yeah.
Okay, well, we just hit the hour, so we should wrap up. So thank you again, both of you, for presenting on theme 1. I'm happy we've gone through all of the themes now with these webinars. So we have one more webinar scheduled for next month. You'll hear more information about that in the coming weeks. And then after that, we'll be on a break for the summer. So it's just one more left to tune in for this season, and then you'll have to wait until the fall. So thank you so much again, and thanks everyone for joining us. If you want to watch this back or share this, it's posted on our YouTube channel. Email me if you want the link for that, but it should be also in your invitation email. That link is there as well. So thanks everyone, and have a great rest of your day.
Thank you, Catherine.
Thank you.
Bye-bye.
Thank you, Catherine.
Bye.
Bye-bye.